Okay, any questions so far on chapter 12? We've been, why? That's a good one. It's a good one. I like that. Okay, so we left off talking about the blood-brain barrier. And this is a selective group of cells. What are the name of the cells? Ependymal cells. And what are they doing for me? Creating cerebrospinal fluid. So remember the neurons of the nervous system in the central nervous system, huge, yes? They're going to need some nutrients. They're going to need some help. Who helps them besides the cerebrospinal fluid? What group of cells are there to act as support? Astrocytes are one of them. What's that whole group of cells called? The glial cells are supporting cells. So one of those, the ependymal cells, is going to help to produce cerebrospinal fluid, and that's what they talk about under the blood-brain barrier. Astrocytes are going to help hold things together with respect to the connection between blood plasma and cerebrospinal fluid production. Because cerebrospinal fluid is an ultrafiltrate of blood plasma. Everybody understand that? Okay, and where does blood plasma hang out? Yeah, in the circulatory system, exactly. So, yeah. So, some substances can pass easily, others are filtered out. And we left off talking about this last class. Certain fat-soluble Certain fat-soluble substances are allowed to pass. Things like alcohol, nicotine, and different anesthetics. Some areas need to monitor directly blood plasma. So we don't necessarily have a lot of cerebrospinal fluid hanging around those areas. Vomiting center, hypothalamus, they have to monitor directly the composition of blood plasma or the chemical composition of blood. So we don't find too much cerebrospinal fluid there, or at least the blood-brain barrier there. Then they talk a little bit about traumatic occurrences, homeostatic imbalances of the brain. Basically, we want a definition of those described in the textbook. Concussion, that's when I bang my head around a little bit too hard in the cranial cavity. A contusion, that's more of a permanent damaged area. I'm messing up some file cabinets in a brain contusion. Subdural or subarachnoid hemorrhage. Subdural, underneath the what? The dura mater. And subarachnoid would be underneath the arachnoid. Remember, there's some spaces there, and there's blood supply there as well. So when we start to have bleeds in those areas, that can be a problem. Forced brainstem through the foramen, that can be a problem. Yes? Shouldn't be there, right? No, it's up further. What goes through the foramen? When we talk about foramen, what foramen are we referring to? The foramen magnum at the base of the skull. So what's there? Yeah, the spinal cord. We don't want the brainstem down there. That can be an issue. Cerebral edema, when I get too much pressure and swelling. Pressure pushes on those neurons and can damage those neurons. It can happen from any type of brain injury. Cerebrovascular accident, what's that? Yeah, that's another name for a stroke. basically we're plugging up the tubes that are going to lead to supplying nutrients for certain areas of the brain. So different different descriptions here. Ischemia, what's that? And that's the one we typically think of when we think of stroke. There's some kind of blockage. So the tissue beyond that blockage doesn't get blood supply. And what's going to happen to that tissue? It's going to start to die. Paralysis on one side, hemiplegia, hemi, can result in sensory deficits. Transient ischemic attack, or TIA, can be temporary episodes, and usually TIAs are going on before a stroke event. We see the two kind of go hand in hand. TPA, what is TPA? That does what? By doing what? Yeah, break up that blood clot. When we talk about blood second semester, we'll talk about some of the different factors involved in creating a clot. And a clot is basically taking soluble proteins and turning them into insoluble proteins. You have to do it quick, though, and it might lead to some other side effects that are undesirable. But the outcome is much, much better for patients who get TPA quickly. I don't know the answer to that question. I don't know if it's defined as that. Yeah, so I don't think it's defined as a stroke or cerebral. I don't think it's defined as that. Not sure, though. Some degenerative brain disorders. You probably all have heard of some of these different ones. Alzheimer's disease, and that is when we start to take those pathways that we keep describing in this chapter, and we start to block them. They're no longer accessible. I can no longer access different file cabinets in the cerebral cortex. So we start with memory loss. Short term is what we see first affected. Some patients with Alzheimer's can tell you stories very vividly of things that happened 30 years ago, but they can't tell you what they had for breakfast. So short-term memory starts to degenerate. Eventually, as the disease progresses, we become more disoriented, language loss, irritability, mood, confusion, all because of all of the different things that are going on, frustrating things that are going on, not being able to access information that was so easily accessed before. Later onset, we can actually start to see hallucination. So patients who are nearing the end stages of Alzheimer's might see someone from their past that's been dead for 40 or 50 years, and they'll start to see those people very vividly. Plaques, and this is something that has been studied for many, many years with respect to Alzheimer's disease. It's a fatty plaque, a beta amyloid peptide that forms in the brain and blocks some of these messages that are being sent to retrieve information in the cerebral cortex. Prions, those are scary. Does anybody know what a prion is? It's a protein, an abnormal protein that actually causes, in the presence of normal proteins to fold abnormally. So it's a protein. It's not a bacteria. It's not a virus. It's a protein that causes damage. That's scary. If you ever want to look at a scary topic, look at prions. Tangles in the neurofibrils can also cause a problem as well. Eventually might kill some neurons off, and the actual size of the brain matter starts to decrease. Another disease, Parkinson's disease. Again, I want you to have a general definition of these homeostatic imbalances in your head. Problems with the basal nuclei, specifically with a group of neurons that release dopamine. And these, again, are ones that are involved in coordination of movement. You know, our friend the cerebellum and midbrain regions, we start to misfire, and pathways are blocked with respect to coordination. We start to see proteins degrade, and again, those pathways start to become confused. Huntington's disease, hereditary, huh? Yep.
Can it be caused by getting hit in the head too much too, though? Because Muhammad Ali has it there? Is that because of being hit in the head so much, or is it just because of what's going to happen?
It probably was going to happen anyway, but it probably came on so early in his life because he got hit in the head too much. So there might be a relationship between head trauma and the early onset of Parkinson's disease. Huntington's disease, also one of the nastier of the brain imbalances. Proteins accumulating. And accumulation, when we think of brain and pathways, proteins and lipids, when they start to accumulate abnormally, think of them blocking pathways, blocking access to information. Initial symptoms are going to be noted in that coordinated movement category again. Later, mental disorientation. We try to block some of the effects of dopamine, and stem cell implants might also be promising. What are stem cells? Why do you've heard that term? Yep, you have stem cells in you too that can become other cells. So when you are first created, how many cells are you? One. Well, how does that cell then become all of the different types of cells that you discuss in tissues and talk about when we talk about different organ systems? Well, the cells have the ability to become different cells. They have the ability to what we call differentiate. And stem cells have the ability to differentiate those immature cells. So if I could take a group of immature cells and cause them to become some of those damaged cells, because you know the neurons you have, that's it, you're not getting any more, right? There's a couple of exceptions to the rule, but for the most part, once they've established themselves as neurons, that's it, you're not getting any more. But stem cells have the ability to be transformed or differentiate into neurons, usually fetal material. So that's all the research and the hubbub about stem cells, because if I can take those groups of cells and cause them to regenerate a group of lost cells in a patient, like a group of neurons, for example, say a patient is paralyzed. If I can cause that pathway to be reconnected, we might be able to regenerate movement in patients. So that's what stem cell research is all about. Mm-hmm. Bone marrow, probably. Yeah, bone marrow. You have stem cells in you, certain cells that can differentiate. For example, when we study the blood, we're going to see that there's one cell that you have in your red bone marrow. And with different signals from different hormones, that one cell can become any of your blood cells. As far as cancer research goes, there's a whole bunch of different research being done on that type of stem cell and others to help regenerate material that's been lost. So that's a tough question to answer. Depends on what the patient is going in for. But it's very interesting research. Yep. because we want to regenerate, we want to get back groups of cells that can regenerate normal cells. Leukemias are groups of diseases, cancers, of those cells, different cell types, overproduction or abnormal production of some of the different blood cell types. And there's a whole bunch of different types of leukemia based on which cell is affected. So yeah, that's what some of that is as well. Spinal cord. What is it for? Yep, so we're getting information from peripheral, bringing it into central, and then we're getting information from central and sending it out to peripheral. What's the next chapter in the book? The peripheral nervous system, yes? So we're leading into our discussion of the nerves, groups of neurons in different nerves that are going to send information to and from the central nervous system. So the spinal cord starts at the beginning, or begins at the foramen magnum. Remember, we don't want brainstem down there. That should be spinal cord. It doesn't go all the way down the spinal column. It ends at the first or second cervical vertebrae, lumbar vertebrae. What does it do? Communication. Information in, information out. There's also some processing that goes on in the gray matter of the spinal cord, and that has to do with reflexes. So when we talk about reflexes and reaction to things very quickly, we do see that there's some processing in some gray matter regions of the spinal cord. So when we look at the spinal cord, we see gray matter and white matter. Remember what the difference was between gray matter and white matter? White matter has myelinated fibers, and typically when we think of white matter, what do we think of? Yeah, travel, information traveling, going from one place to another. Gray matter, unmyelinated fibers, and who? Yeah, cell bodies. So when we think of those parts, think of processing. Can things go in gray matter? Yeah, they tend not to go as fast because of the unmyelinated fiber. So they usually go short distances, or they go to access information that's being processed in nuclei. So protection. What's the protection of the spinal cord? Well, we have meninges, just like we do in the brain. What are the three meninges that are going to help protect the spinal cord? Dura mater, arachnoid, and pia mater. Yes? They run all the way down the spinal column as well as cerebrospinal fluid. So again, we're going to find all of this stuff throughout the entire central nervous system. What's an epidural space? Anybody ever have an epidural? No, that's intervertebral. Go ahead. Yes. Yeah, it's a little group of lipid material that acts as a cushion. And why do you think we need a cushion? Yeah, you won't be slamming up against the bones, exactly. That's going to damage the cells. Cerebrospinal fluid are in the subarachnoid spaces, and the dural and arachnoid membranes are going to extend all the way to the sacrum. Now, you should know what I'm talking about now, because next week, what are you going to be doing? You're going to be taking your second lab exam on bones. So you're a pro at what the spinal column consists of, correct? What does it consist of? Bones like 7, cervical, 12, thoracic, 5, lumbar, well, that's the spinal column. What's at the bottom? Sacrum, coccyx, yes? Now, lumbar puncture. Did they show us the little picture in the book? Oh, yeah. On page 466, figure 12.27. Have you ever had a lumbar puncture? Ladies, some of you delivery ladies, opt for the epidural. Amen is right. I was so bummed out with number one. It's like, no drugs, no drugs. Stupid. Number two is like, give me the drugs. And they're like, no, sorry, too late, time to push. So I never had the pleasure. Pretty upset about that. But if I'm going to put some drugs down there what does an epidural do for me? Yeah, it deadens some of those pain receptors receiving information lower than where you inject, right? You know, lumbar, vaginal region. You see why that works? But you don't want to put a needle into your spinal cord. Why would that be a problem? Yeah, you could cause a lot of damage. So remember, the spinal cord proper ends around L1 and L2. So where, if you look at this picture closely, for those of you who have a textbook, oh, I have it on here. Where is that needle being inserted? Yeah, way below L1 and L2. Because remember, we're not all built the same, correct? Yeah, we want to make sure we're way, way down. What if your lumbar vertebrae, or what if your spinal cord ends around L3? Oh, it was too bad, so sad for you. Yeah, that wouldn't be so good. So we go lower in between the intervertebral discs, and we can inject material that's anesthetic into the spinal column to deaden some of those nerve groups that are feeding information in and out. In and out. Can you move your legs? Nope. So motor and sensory, exactly. And we want to make sure that we don't damage the spinal cord in any way. Also, if you want to take up some fluid and analyze it, cerebrospinal fluid, you can analyze it in the lab and look for some chemical imbalances to see if there's different diseases going on. So, where does the spinal cord end? That awesome diagram on page 465, you will probably at some point see again. I'm guessing on your third lab practical exam. But where does the spinal cord end? What's the end called? Yeah, the conus medullaris. And then coming out the bottom are basically different groups of nerves, just like wires in your house, that are feeding into the bottom of the spinal cord. That group of nerves feeding in are called the cauda equina. Because if you look at them, and you can see the pictures on page 465, especially the one at the bottom, D, you can see it kind of looks like hairs or a horse's tail. What's cauda equina mean? Horse's tail. And that's what it looks like at the bottom. Some of the different things that hold it in are some ligaments and tacked in places so that the spinal cord doesn't bungee up and down. Would that be good if it bungeed up and down? No, because think of all the wires or groups of nerves that are coming in through the intervertebral discs. And of course, you've seen that on the bones when you examine them in lab. You see all those little holes? Some of those are also nerve groups coming in, not only blood supply. So if the spinal cord inside that case moved up and down, what would happen to all of those wires feeding in? They would pinch and snap, and that would not be good. So we need something to hold it down. The filum terminale extends all the way to the coccyx region, and that's an extension of the pia mater that acts as a tack to hold everything in place. It's going to help anchor the spinal cord. Again, we see a spinal tap or some kind of lumbar puncture. And this diagram on page 465 shows us some of the different areas of the spinal cord. So we have our different vertebrae: cervical spinal, thoracic spinal, and lumbar spinal, and sacral spinal nerves are going to feed into the spinal cord in those regions. So if I asked you to point to your cervical spinal nerves, where are you going to point? Into the neck region. What do you think feeds in through the cervical groups or cervical plexuses? Yeah, everything kind of in that region, yes? So we see that these groups of nerves are, and we'll talk about them in a minute, are very closely related to the areas in which groups of nerves feed information into and out of the spinal cord. 31 pairs of spinal nerves is what we have. And they are all part of the peripheral nervous system, PNS. So we're going to talk about the specifics in chapter 13. We see that there's a whole bunch that feed into certain areas of the spinal cord. So if you look at the spinal cord and the thickness of the spinal cord, we're going to see in the cervical region and in the lumbar region, the spinal cord gets a little fat because of all the information coming in. Think about what's coming in the lumbar region, lower lumbar region. A lot of information from this whole area here, the abdominopelvic. And what's going on in there? We got the digestive system, we have some reproductive organs, we have the kidneys, we have the spleen, we have the liver, we have the pancreas. You getting the point? A lot of stuff going on in the abdominopelvic region. Cervical region, a lot of stuff that's feeding from upper, and the arms are feeding into those areas. So we see an area called the cervical enlargement and the lumbar enlargement. The cauda equina, collection of a bunch of things coming from the lower limbs. So think about all the muscles that you have to innervate in your lower limbs and in your hip region. We see those roots coming at the bottom of the lumbar region of the spinal cord. Cross-sectional anatomy. If I cut it in half, and the nicest way to see this is on page 467 and 468. So there's a lengthwise groove or fissure that sort of separates the spinal column into two distinct halves. Not all the way through, but pretty deep. There's a ventral one, which is what? Ventral is what? Front. Dorsal is back. So ventral anterior, we see one called the median fissure. And dorsal or posterior, we see a lesser of a groove. We call that a sulcus, called the median sulcus. Inside that gray matter is connected by an area called the gray commissure, and we're going to, again, do a little bit of processing in the spinal column. So when I look at this, what's anterior and what's posterior? Remember, it's the spinal column. So if I hold a vertebrae, what's the front part? The body of the vertebrae. And what's the back part? The spinous process. So you see where anterior is? So we see the fissure anterior and the sulcus posterior. The spinal cord is in there in the center. We see those little yellow things feeding in. What are those yellow things feeding in? Yeah, those are groups of neurons. A nerve is a whole bunch of neurons. When we talk about muscles, we're going to see some of the same terms referring to nerves as we do to muscles and their arrangement. So what we're going to see are different areas, different pathways, different groups of gray matter, different groups of white matter. Again, when you become the brain surgeon, you're going to have to know what all these different pathways do. But for my purposes, I'm not going to make you memorize all of the different pathways. Aren't you glad about that? But I do want you to know basically what these areas do for a living. So when we talk about dorsal horns, and did you see the shape of the gray matter within the spinal column? It's got little projections or horns that come off. Dorsal, ventral, lateral, all different pathways of material coming in, going out, being processed. Dorsal horns. So this is what you need to know. Gray matter dorsal horns are for what? Somatic and visceral sensory input. What the heck is that? Correct. Somatic, body, soma, and visceral would be your internal organs. We're going to get sensory information coming in, dorsal. We don't want to cross paths with information, right? We don't want things coming in and out in the same pathway, right? We'll have a head-on collision. We don't want that. So we have a different horn, ventral horn. What's ventral? Front. We see some interneurons there. These are the guys that do what? Or they, yeah, exactly. They're going to hand off. They're going to connect between input and output. So we see some somatic motor neurons, axons that exit the cord via the ventral root. Now on the side, lateral, and that's only in the thoracic and the beginning of the lumbar region, we're going to see sympathetic neurons. What the heck is a sympathetic neuron? So sorry. Anybody know? I don't think we've talked sympathetic, parasympathetic yet. We'll talk. Has to do with the, you're right. It has to do with the nervous system. Cookies. Think my cookies. Running to get my cookies. What do I have to do in order to get more blood flowing and get more oxygen to my... I have to do what? Speed stuff up, yes? Sympathetic divisions of the autonomic nervous system in the peripheral nervous system, that was one hell of a mouthful, wasn't it? Are going to help do what? Speed up responses. So we see sympathetic neurons here in the lateral horns. Dorsal horns, sensory input to the cord, dorsal roots. And dorsal roots or spinal ganglia are groups of cell bodies that are sensory neurons in nature. And we're going to see them kind of hanging out a little bit further from the spinal cord proper. So, on page 468, figure 12.29. This is a nice one because I like the whole visual thing and I like the colors. So if you stare at that long enough and then you shut your eyes, hopefully you'll see it again. But this is a map of the gray matter. So we see somatic sensory, SS. Where is that? Dorsal. Visceral sensory. Still dorsal but closer to medial regions. Visceral autonomic motor, or VM. What's autonomic mean? Automatic. Yep, that's going to be in those central regions more towards the ventral horn. And then somatic motor is going to be ventral. So sensory, dorsal, motor, ventral. Yes? So this diagram is going to help you remember that, hopefully. And then you can see all the different pathways of information coming in and out. We see that a nerve, a group of neurons, splits just before it comes into the spinal cord. And this is where I'm going to start to make sure that pathways are followed properly go to the right place, go up the right street to get to the right file cabinet. And this is where we start splitting off information, right as the spinal nerves feed into the spinal cord. So the ventral root tends to be motor, the dorsal root tends to be sensory, and we see how a nerve contains what? Are all nerves just containing one type of neuron that goes in one direction? No. Most nerves are a combination of information coming in and information going out. A combination of information that'll split and go to the ventral root or split and go to the dorsal root. What protects them from firing each other off? Yeah, myelination. So we see another discussion of that here. Myelinated and non-myelinated nerve fibers allow for communication between parts of the spinal cord. So we're going to find a lot of those non-myelinated guys in what portion of the spinal cord? Gray matter. So we see that these pathways are going to run in three different directions. They're going to go up, they're going to go down, and they're going to go side to side. So ascending, descending, and transverse. Again, following different pathways, we're going to see different maps. And this, on page 469, figure 12.30 shows us some of those pathways outlined for us. So white matter is divided into three white columns or funiculi on each side. Again, dorsal, lateral, ventral. Each tract has its own little axons and has a very specific destination and a specific function. Decussation, pathways that cross to the other side. Relay consists of 2 or 3 neurons handing off. So we might, in a reflex, for example, get sensory handing off to interneuron, handing off to motor neuron. Somatotropy, precise spatial relationship, and symmetry, pathways paired symmetrically. So all of these are different setups that we're going to find in the spinal cord to relay information to where it needs to go. Again, I'm not going to make you memorize all of these, so don't panic. There's a lot of them, and it's scary. But the nice thing about this diagram, it shows us the map of the spinal cord and some of those different pathways mentioned. So ascending tracts tend to be where in the spinal cord? What's front and what's back? So it's going to be dorsal, right? Because look at the front. The front has that big fissure in it, right? Dorsal's in the what? In the back. So most of those ascending tracts tend to be in the back of the spinal column. There are some laterally, and we see them outlined in blue as well. Very few of them are going to run up the center. When we're talking about descending tracts, those tend to run more towards the center of the spinal column, and those are outlined here in the diagram. Okay, so again, I'm not going to make you memorize all the different pathways, but you should understand the difference between ascending and descending pathways and where I'm going to find them on the spinal cord. Okay, does everybody get what I just said? Yes? Okay, good. So when we talk about things going up, ascending pathways, there's 3 different groups of neurons that can help that information go for cerebral cortex processing. We have a group of neurons called the first order neurons. Those are the guys that are going to send information from cutaneous receptors and proprioceptors. What the heck are those? Skin and what? Those guys that are going to help me keep my what? Balance and equilibrium, proprioceptors. There's some in the head. There's also some in the muscles as well. They're going to re-enter the spinal cord, and then they're going to have a little bit of a split over in the medulla region, and they're going to hand off to the next group of neurons, which are called what? Second-order neurons. These guys are usually referred to as an interneuron of some kind or another. Cell body in dorsal horn of spinal cord. What the heck does that mean? Dorsal horn of spinal cord. Yeah, that gray matter picture, the dorsal end. And axons extend to the thalamus or the cerebellum. Again, this is the first place where we're going to start getting that information to where it needs to go for processing in the cerebral cortex. Then we're going to hand off to what? A third-order neuron. Again, also referred to as an interneuron. Cell bodies tend to be in central brain regions like the thalamus. The axons extend to the somatosensory cortex. Remember what we talked about when we talked about different areas of the brain. What was the somatosensory cortex going to help then relay information to? Anytime we see the word cortex, we're talking what? Yeah, the cerebral hemisphere, some portion of the cerebral hemisphere. Remember, I have to access information from certain file cabinets, certain stored information to perform different functions. So the somatosensory cortex is going to help relay that information, to access that information for processing. Cerebellum has no third-order neurons. Cerebellum is only going to then send message directly from from a first-order neuron to where it needs to go as far as coordinating movements. So, three main pathways, ascending pathways. We have the dorsal column-medial lemniscus pathway, the spinothalamic pathways. These are going to help with what? Yeah, touch and, yeah, when I start to fall, I catch myself. Or I'm walking down the stairs or walking up the stairs. Believe it or not, I have to change what I'm doing with respect to information going to my muscle groups. And visual keys or cues are going to help me with that as well. So these guys are going to help with sending that message to those specific places. And then the Spinocerebellar Tracts are going to end in the cerebellum. When I touch something, do you ever do that at a science museum with your kids or do it yourself? You stick your hand in something and you touch it. You can't see it. You're getting no visual cues. You're getting no cues from smell, but you can a lot of times figure out what it is. How come? Because of these pathways. Because that's another part of learning things, touching. So I can tell it's an orange, or if somebody gets gross, you can, put spaghetti and grapes for eyeballs and stuff like that. Since we had Halloween, you've probably done that with your kids too. A little torture. So we see different pathways to the brain, medulla, and the thalamus that help with discriminating certain things, touch and vibrations as well. Again, this diagram on page 470 in your textbook is scary, yes? Don't be scared. Well, I'm not going to make you memorize it. But we can see information coming in following some of these different pathways for processing in the cerebral cortex. So we have to take different roads to different places. We see the spinocerebellar pathway and the dorsal column-medial lemniscus pathway, and how they can actually cross over for processing in the cerebral cortex. So, spinothalamic pathways, lateral and ventral, they're going to transmit information about what? Pain. Also temperature. Can temperature be associated with pain?
Yes.
Yeah, if you've ever been out in the cold way too long. We are going to do a series of torture-like labs, which I always enjoy, and we will elicit pain in you after we have our skeletal system test. Yeah. So we'll make you stick your hand or your elbow in really, really cold water or really, really hot water, and we'll see what happens. So transmission of pain, temperature, coarse touch, pressure, all travel these pathways. Spinocerebellar tracts to the cerebellum. And remember, when you think cerebellum, think what? Coordination of muscle activity, some kind of coordinated movement. So think coordination and skeletal muscle. Again, we see some of those pathways as well. Now, descending pathways, what are those? Coming down, going where? When you think coming down, down and out. So think motor. So delivers impulses from the brain to the spinal cord, so we then can deliver a motor response to the proper area outside the spinal cord. Pyramidal tracts are direct pathways, and then we also have indirect pathways as well. So motor pathways involve two different neurons. We have an upper motor neuron and a lower motor neuron. The upper motor neuron is going to get information from upper regions, primary motor cortex. And the lower motor neurons are then going to send message where? Now we're talking motor, so are we dorsal or are we ventral? We're ventral, so we're going to send information out the ventral motor neurons, ventral horns, to where? The skeletal muscle. So upper coming from brain, lower going out to innervate. Everybody knows what I mean by innervate, right? Cause something to happen, stimulate something. One motor neuron can stimulate another motor neuron, or it can stimulate a muscle to contract, or it can stimulate, what's a third thing a neuron can communicate with? Another neuron, a muscle, or a gland to secrete some sort of chemical. So we have direct pathways. Axons are going to synapse or connect or communicate with interneurons or somewhere in the ventral horn, motor neurons. Direct pathways are going to regulate things fast and fine skilled movement. So if I have to move quickly, some of these direct pathways are going to be involved in that movement as well. Similar to what we saw when we discussed the reflex. So lateral and ventral corticospinal pathways. Again, I'm not going to make you memorize these, but we see their travels up the spinal column. Indirect, multineuronal system. Think the connecting guys. Brainstem, motor nuclei, groups of neurons that are going to process and relay motor information. And all motor pathways except pyramidal pathways are going to go through this indirect or multineuronal system. So, pathways are going to help regulate things like axial muscles, maintaining balance and posture. When I'm just sitting here, even though I'm not moving through space, if I wasn't able to control certain muscle groups, what would happen to me? I would fall over. I wouldn't be able to keep my head up. So that's the groups of muscles we're talking about. coarse limb movement. When we talk about muscles and we talk about movement of limbs, it's not just one muscle that's going to cause movement to occur. When I move my arm like this, which muscle's doing most of the work? This guy, the biceps, but is that the only one? No. We have this guy over here, the triceps, who has to stretch. Then we have lateral muscles that are going to control the movement of my arm so that it doesn't jiggle around at the elbow. Yes? So all of those muscles, axial muscles, and coarse limb. Head, neck, eye movement that follows objects in their visual field. All of these are indirect or multineuronal systems. Oh gosh, don't worry about those.